talked about mitosis. So today it's all about meiosis. Hello and welcome back. Meiosis is a specialized form of cell division which takes place in sexually reproducing organisms. So in humans, for example. Meiosis is in general a key feature of eukaryotes, as it generates greater genetic diversity. And therefore it's important for sexually reproducing organisms, because that's the biological reason behind sex. Its origin, however, is poorly understood, even though it's observed in all eukaryotes, so it's also in all animals, in plants, in fungi. Within humans, our chromosomes are divided into 4 to 4 autosomes and 2 sex chromosomes. We talked about them in the DNA video, which is linked above. These sex chromosomes, so X and Y, which determine our sex, they have a certain feature, which makes them special. They are haploid. Haploid means that in every cell there's only one copy of the chromosome present, and not two, which is called diploid. To be haploid is crucial for egg and sperm cells, as otherwise in every replication round, the total number of chromosomes would double. And that's not what we want. We want 46 chromosomes. The process itself, or the phases, are similar to mitosis. So if you haven't seen this video, watch it now. The first step is to separate the sister chromatids, but the main difference here is that the homologous chromosomes get separated as well. So we've got meiosis 1 and meiosis 2. The phases are called the same as in mitosis. So we've got prophase, metaphase, anaphase, and telophase. There are two main differences. In prophase 1, there's a process called homologous recombination, where the two sister chromatids cross and exchange genetic material. And this is important because that's what we want. We want genetic variation. The parts of the chromosomes where they overlap are called chiasmata. In human fetal oogenesis, and that's a process where women start to produce eggs, which actually start when we're still in the womb of our mothers. Then meiosis is arrested at the end of prophase 1 and starts again when a girl hits puberty. So that's known as the menstrual cycle. This arrest is not observed in males, as males only start to produce sperms when they hit puberty, and usually they can do that until they're dead. So these differences are of course essential, but apart from that, the processes itself are quite similar. So in metaphase, the chromosomes become more condensed and they start to arrange in the metaphase plate. In anaphase, the homologous chromosomes are separated, but not the sister chromatids are pulled apart. And the cells are separated in telophase. So that's the difference. We're not pulling apart the sister chromatids, but the homologous chromosomes. The sister chromatids are getting separated in the second meiosis. Between meiosis 1 and meiosis 2, there's no duplication of the genetic material. That is important because we want to have a haploid genome afterwards. So no replication of genetic material. So during meiosis, it's the same process again, but now we're not pulling the homologous chromosomes apart, but the sister chromatids. So the result of this process is the formation of 4 haploid cells. So we're not getting 2 as in mitosis, we're getting 4. And they all contain only one homologous chromosome, and they all have just one chromatid. In humans, as I mentioned before, these cells are called egg and sperm cells, and they join in fertilization to form a new human being. The annotation is as follows. 2n, 4c gets to 1n, 2c gets to 1n, 1c. The number, so n stands for the number of homologous chromosomes, and n stands for the number of homologous chromosomes. So we first got 2, and then we got only 1, and the c is for the sister chromatids. So we've got first 4 sister chromatids, then only 2, and then just one sister chromatid. Two sister chromatids are called diploid and one haploid. I hope you enjoyed this episode and see you in the next one.